So in the dicot plants, here the embryo is having two cotyledons. So it is the embryonal axis. The embryonal axis, one end is with plumule. Embryonal axis ending with plumule, or this plumule is developing into shoot system. So plumule develops into shoot system. And it is the radicle. The radicle develops into root system. And these are the cotyledons. Cotyledons, and the cotyledons are rich in nutrients or proteins. In pea plant or Fabaceae members, Leguminaceae family members, the cotyledons are rich in proteins, or they are storing the proteins. And here plumule, radicle, and two cotyledons completely is embryo structure. And in the here plumule and radicle, these are not covered by any sheaths. Here plumule is developing into shoot system, radicle is developing into the root system. Here two cotyledons are present, and these two cotyledons are rich in nutrients, or sometimes proteins are present. Here endosperm, in the generally seed, here endosperm is also present. So endosperm is the result of a double fertilization. In angiosperms, here double fertilization is observed. Double fertilization means two fertilizations. So due to the first fertilization, zygote is formed, zygote develops into embryo. And due to the second fertilization, here the endosperm is developing. Endosperm is here sometimes in the seed endosperm is present, sometimes it is absent. If the endosperm is present, the type of seeds are called as endospermous seeds. And if the endosperm is absent, non-endospermous seeds. Endosperm is present in some seeds, like in castor. In castor plant, endosperm is present. So here after the development of the embryo also, some endosperm is remaining. So the remaining endosperm is, it is if the endosperm is present in the seed, that is called as endospermous or albuminous seed. And non-endospermous seeds, generally dicot seeds are, they are not storing the endosperm. Non-endospermous seeds, the endosperm is completely utilized after the maturity, after the development of the embryo and seed, endosperm is completely utilized. So if the endosperm is completely utilized, there is no endosperm in the seed. So here these are pea seeds. So these are some examples, non-endospermous seeds. Endospermous, non-endospermous. So it is about structure of the dicot seed. Two cotyledons, embryonal axis, plumule, radicle, and in seeds sometimes endosperm is present, sometimes absent. If the endosperm is completely utilized, no endosperm is present, like in pea plant. And sometimes endosperm is not completely utilized. Some amount of endosperm is remaining. So example, castor. Let us discuss about here structure of the monocot seed.